Hello and welcome back to biology. So today we're going to be talking about cellular respiration, kind of start to dig into this process a little bit. So cellular respiration, what it is the process on how we get energy, right? And on how we make energy. So all it is it's a, whoops, missed one. It's a set of reactions, right? So it's really cellular respiration is a big part of your metabolism. Is what we talk about. So it's a set of reactions that occurs to convert to make ATP, because ATP is this is that usable form of energy that we talk about. So they do it to make ATP, so that way we can have energy, because ATP is the form that our energy has to be in order for us to use it. And this process occurs, or at least part of it occurs, in the mitochondria, right? So what are we talking about here? Well, this is the general reaction that we talk about for cellular respiration. We take glucose plus oxygen, and we're going to make a little bit of carbon dioxide, a little bit of water, and then some energy. And that energy is in the form of that ATP. Now, we make a distinction between heterotrophs and autotrophs. Heterotrophs are organisms that we have to consume other things to get our energy. Because really what we're doing is we're taking the energy that's stored in glucose, and converting that into ATP. Right? So for heterotrophs like us, we have to get our glucose from eating things, plants, other animals, that idea. Autotrophs, on the other hand, make their food, right? That's the key part there. Is autotrophs make glucose. So autotrophs are going to make this glucose here by capturing energy from the sun, and then with some carbon dioxide and water, and they're going to turn it into glucose. Now here's a very important distinction to make. Plants, because this is basically photosynthesis here. Plants will also do this process, right? Because plants, same as us, same as every living thing out there, cannot use glucose as a source of energy. Now it's great for storing energy, but then for them to use that energy, they also have to convert it to ATP. So when we talk about photosynthesis, and really when we talk about cellular respiration, every living thing, us to fungus to mushrooms to plants, we're all going to do this process, or at least some form of this process. Now I say some form of it because there's two different types of cellular respiration. There's aerobic respiration, which requires oxygen, and then there's anaerobic respiration that does not require oxygen. So I like to think of it as like cellular respiration is like this flagship term. And then if we have oxygen, we're going to go to the aerobic pathway. If we don't have oxygen, we're going to go through the anaerobic pathway. But we're going to start off by talking about aerobic, or really part one. So part one, so there's 3 steps to respiration. Glycolysis, the Krebs cycle, you may also hear it referred to as the citric acid cycle, and then the electron transport chain. Glycolysis is what we're going to talk about today. So glycolysis is step 1, no matter if you're going to go aerobic or anaerobic. It's step 1 either way. So let's get right into glycolysis. So glycolysis, if we break down the word, glyco, which stands for glycogen, it's basically glucose, it's a sugar. And this means splitting. So what happens during glycolysis is we're literally splitting carbon, we're splitting glucose in half to form 2 3-carbon molecules. And those 3 carbon molecules we call pyruvate. So glucose gets split in half, so we go from 1 6-carbon molecule to 2 3-carbon molecules that we call pyruvate. And throughout this process, we're going to get 2 ATP and we're going to get 2 NADHs. And we're going to talk about that. So our products here. So you don't have to memorize the steps. There's like 10 steps turning glucose into pyruvate, all right? What we need to focus on are our inputs and our outputs. So our input is glucose. So this is what we put, this is what we put into the process, is this glucose here. But then our outputs are ATP, 2 NADH, NADs, and 2 pyruvates. So when we talk about the ATP real quick, we talk about net ATP because in order to break glucose in half, we have to spend some energy. So if you look here, like this is what we're doing over here. We're spending some energy. We're spending some energy to break glucose. And when we break glucose, we actually get 4 ATP out of the process. But since we put in 2 to get 4, we really only we only gained 2 ATPs. The other thing is this NAD over here. So NAD, there's 2 different forms. There's NAD plus and NADH. NAD, we're talking about NAD and FAD, but what they are is they're energy storing molecules, because whenever we break a bond or break apart a compound, we release energy. And we're going to lose energy throughout this process, but we want to try and conserve as much energy as we can. And that's what NAD comes into play at. NAD is going to try and conserve, it's going to kind of take in some of that energy to help conserve it so we don't lose a lot of it. So NAD plus is the low energy state, so it's not carrying anything. It's just chilling, right? And then NADH is the high energy. So NAD plus is the low energy. NADH is the high energy. Oops, I don't know why I keep doing that, but so NAD plus is low energy, NADH is high energy. So we made 2 NADHs throughout this process, 2. Those NADHs, we're not going to do anything with them right now. They're going to come back during the final stage, during the electron transport chain. They're also used in anaerobic respiration, but I'll save that for the end. So input for glycolysis is we input glucose, it gets cut in half, giving us 2 pyruvates. We also get 2 ATPs, 2 NADHs from this. So glycolysis has been happening out in the cytoplasm, okay? This process of glycolysis has been happening out in the cytoplasm, okay? Now, if it's anaerobic, we stay in the cytoplasm. We stay in cytoplasm. But there's aerobic, if it's with oxygen, then we move into the mitochondria. And the other 2 parts of this, the Krebs cycle and the electron transport chain, take place inside the mitochondria. So as part of this, let's just talk briefly about the mitochondria. I just want to talk through just some parts of the mitochondria. So when we're talking about it, you kind of know what we're referring to. So this part of the mitochondria here, kind of this big outer part, is the outer membrane. So it's the outer part of the mitochondria. Well then, since we have an outer membrane, we have to have an inner membrane. Yes, very good. So that's what this does, all these yellow lines here. This is the inner membrane. Inner membrane. Nice, good. And then inside the inner membrane, you see like all this blue space here. All this blue space is, we call it the matrix. Just like the movie, just like Neo, right? It's the matrix. That's happening a lot more. Matrix. And the matrix is actually where the Krebs cycle is going to happen. So the Krebs cycle is going to happen in the matrix of the mitochondria. And then you see the inner membrane is not folded nicely. It's all hickly-pickly. It's all folded on top of each other and trying to maximize as much inner membrane space as possible. So we call all these folds here, like all these folds like right in here. Well, that is the cristae. Call them cristae. And that's where the electron transport chain is going to take place. It's going to take place in the inner membrane. or across the inner membrane here. So that's why that's why we have all the folds in there, so that way we can get as much inner membrane as possible. So in that way we can have the electron transport chain happening in a lot of different places. So that's it for this video. Let me know if you have any questions. I look forward to seeing you in class, and I hope this was helpful. Bye.